of replication, basic concept of recombination, and little bit of PCR. We did couple of things. That DNA replication is semi-conservative. DNA replication is semi-conservative. It's bidirectional. Replication requires unwinding of the DNA helix. DNA replication is semi-discontinuous. Correct? All these statements make sense. Lagging strand is formed from the Okazaki fragment. So let's review what we did. So the Watson and Crick said that there is two helix of DNA, antiparallel, correct? And each of the strands of the helix acts as template for the synthesis of the new DNA, complementary DNA. And this is what exactly demonstrated by Arthur Kornberg in 1957 by discovering the DNA polymerase 1. That's the first DNA polymerase discovered. And using the DNA polymerase, he says that you can replicate the DNA inside the test tube. All you need is a template, a primer, correct? You need DNA polymerase, DNA polymerase 1, and you need nucleotide triphosphate and some metabolite, correct? This is what he demonstrated, and he got no result. So, the nucleotides are added at the 3' end of the primer, correct? Always 3' hydroxyl group has to be free so that the incoming nucleotide can form the phosphodiester bond and 3' end of the primer. All one catalyzes something. There is something called processivity. It's a very important term in terms of enzyme kinetics. Processivity means how good the enzyme in case of DNA polymerase, how many rounds of DNA nucleotide incorporation it can do without dissociation. More processive the enzyme, it can go longer. So they get associated more. Does it make sense? So that's called processivity. So about 20 cycles for pole 1. Pole 1 is about 928 amino acid long, and we already saw it last class that if you 59, you separate this 1 to 328, correct? 324, and the C terminus, which has the polymerase activity and proofreading activity, is called clino fragment of DNA polymerase. Correct? We discussed that. Now, lagging strand, leading strand synthesis is continuous. You start with one primer and just keep on synthesizing complementary until you are terminated at the end. Does it make sense? So it's a continuous process. There is a lagging strand because of the direction. Correct? Because of the direction issue, this is 3' end. So nucleotide incorporation can take place here, but your polymerase is here. So to avoid that, to put the 3' end of the primer close to the active site of the DNA polymerase, this forms a loop. So that 3' end of the primer is facing the active site of the DNA polymerase, and in that process, it starts to produce short fragments of new DNA, which we call Okazaki fragments, correct? And these Okazaki fragments, as the polymerase moves forward, these Okazaki fragments are complementary and left on the lagging strand. Eventually, these Okazaki fragments needs to be ligated to form a continuous form of DNA, and who does that job?
Ligase.
So total processing of the Okazaki fragment, what are the enzymes you need?
DNA ligase.
DNA ligase is the last one to connect.
Polymerase one.
What is before that?
Polymerase one.
You need DNA pol 1, and pol 1 has a specific activity. Which one is needed? Exonuclease is true, but you have to be more specific. Is it 5' to 3' or 3' to 5'?
5 prime to
5' to 3', because your primer is at the end, 5' end of the Okazaki fragment, correct? And this primer is a RNA primer. RNA primer is supplied by the primase, correct? So you need to degrade this RNA primer by 5' to 3' exonuclease activity. Then what? Then you have a gap, correct? What needs to be done? Gap needs to be filled with new nucleotide incorporation, so which is also done by pol 1, is called nick translation or gap filling. And once the gap is filled, RNA is removed, gap is filled, you attach two ends of the DNA, so you ligate. So that's how the Okazaki fragments are ligated, and the polymerase 1, pol 1, does this particular job. So how many enzymatic activities present in pol 1?
3.
2 or 3? 3. It has, what are the 3 activities? Pol 1 has the polymerase activity, correct? Where it incorporates the nucleotide. What is the next activity? It has 2 exonuclease activities, 3' to 5'. Exo, what is the function of this? Proofreading, correct? And you have another activity, 5' to 3', exo, which we just discussed. What is the function?
Removal of the RNA primer.
It's removal of the RNA primer from the Okazaki fragment, correct? So these are the 3 different activities it has. All 3 so this is a functional replication fork, but along with the polymerase, pol 1 is involved in the Okazaki fragment, correct? Ligation, not ligation, I mean the processing. And the pol 3, DNA polymerase 3, is actually the replicating polymerase. There are 2 DNA polymerase. So there are 2 pol active sites in 2 different DNA polymerase, correct? One is helping the replication of the leading strand, another helping the replication of the Okazaki fragment. Does it make sense? So along with these, so this is polymerase 3. Polymerase 3 is the actual enzyme which helps copying the genome in E. coli. Does it make sense? While pol 1 does help the processing of the Okazaki fragment. What is the function of pol 2, pol 4, and pol 5? There are at least 5 different DNA polymerases. Normally, the function of the pol 2 and other polymerase is in the repair. So if there is a DNA damage, you remove some part of the DNA, correct? And then you fill it up and repair, and those are the situations where those polymerase works. So along with the polymerase, you need an helicase to open the helix. You have to make single stranded DNA. single-stranded DNA is stabilized by single-strand binding protein. So once the DNA is opened by the helicase, until the replication is complete, correct, it has to be in the form of single strand. So that's why this single-stranded DNA is critical because they stabilize the single-stranded form. Then you are opening the helix, so you are putting a lot of supercoiling stress, correct? This supercoiling stress is released by this gyrase, cuts the DNA and again this is. So then you have the primase which supplies the RNA primer for the synthesis of the Okazaki segment. So is it clear the whole function of the replication fork? Yes.
Are helicase and primase the same?
No. Then
So,
yes.
is topoisomerase and DNA gyrase the same?
They have different functions, but topoisomerase also can change the topology. Okay, so polymerase 3 is the real polymerase in E. coli. It's a multi-subunit protein, but we are not getting into detail. At least 10 different subunits present in Pol3. Now that's about prokaryotic replication. We briefly touched the eukaryotic replication, of course, much more complicated, correct? But the chemistry is identical. You have a nucleotide incorporation using the polymerase active site, you have the proofreading, you have the Okazaki segment, all these things same in eukaryote. But eukaryote is much more evolved, so it's more complex, correct? There are a lot of other proteins, a lot of other functions. So there is 4 different phases of the cell cycle. These are the 4 different phases of the cell cycle. I think we briefly touched the G1, S, G2, and N. G1 is called the gap 1, correct? And S is the phase of the cell cycle where your DNA replication takes place. That means DNA is duplicated in this S phase of the cell cycle. Then G2 is the gap 2, mitosis is the place where your DNA, duplicated DNA, is separated into 2 daughter cells, correct? Segregated. So the gap 1 or G1 phase actually is when the 2 daughter cells are produced from mitosis. These cells are tiny cells, small, correct? They have less protein, 1 copy of DNA, correct? Eventually, they also need to be duplicated. They also need to divide. So these cells need to grow. Grow means what? Make protein, correct? Transcription, translation, so that we have more protein, enough proteins for making 2 cells. So basically, in this case, lot of transcription and translation takes place. Once the cell is grown enough, then cell thinks that it's ready for division, not division, ready for replication, correct? Prior to division, cell has to duplicate its genome. And this is the S phase. So it's very important that there is a boundary between G1 and S. Unless the cell is ready, it will not go into S phase. If there is plenty of mutation in the DNA, it's not going to go into S phase, because S phase, it will just replicate the DNA. If you have mutated DNA, it won't replicate, because we will be developing daughter cells with mutation. Does it make sense? So those checkpoints are heavily controlled. It's a very important area of study, how exactly the cell cycle checkpoints are regulated. There are many proteins which controls the cell cycle checkpoint regulation. I think I mentioned one protein last class which controls the G1 S-phase transition. What is the name of that protein? P53. P53 is a tumor suppressor. One of the most well-studied tumor suppressor. Correct? It's one of the most widely studied tumor suppressor because if this particular protein is mutated, then there will be cancer. Or most of the cancer has the P53 mutation. One of the function of the P53, we don't go into detailed signaling further here, is that it's a police officer to make sure that, correct, the DNA which is going to be replicated there is no error. If there is a mutation or DNA damage, P53 will halt the cell cycle. Okay? Stall the cell cycle not to go into S phase. Okay? It safeguards the genome. That's why if P53 is mutated, so there is no really checkpoint person, correct? So mutated DNA gets into the S phase and duplicated, and cell will replicate the mutated genome. And if there is plenty of mutation accumulated at some point of the cell cycle, it will lose the control on cell proliferation. There will be abnormal cell proliferation, oncogene activation, and cancer. Does it make sense? So P53 mutation is that's why it is often found in hereditary cancer. What causes mutation? We will talk about today. Correct? We'll talk about repair and all cancer. So I gave you an example, but it's not only P53. There are many other proteins. Some of them we know, some of them we do not know. Correct? That's why if we take the cells and if we take the cells, what I wanted to say, I forgot. I lost my thought in some other place. So, yeah, if we expose to stress like UV light, correct? UV light has a lot of these ultraviolet light, or if you take the sunbath also has some amount of UV light, and that damages DNA. If you stay in one week in Florida Beach, correct? You will be tan and all the skin will be peeled off because cells are dead. So normally our physiological system has the mechanism to take care of the damaged cells. But some people who have less ability to repair or take care, they will have problem in handling these UV radiation. So if you have a P53 knockout mice you can knock it out a particular gene. So this particular mice will be very sensitive to UV radiation because it has repair system is not working properly. Does it make sense? So repair is very important, and these cell cycle checkpoints are heavily correct, regulated. So once the DNA is replicated in this phase, there are some transcription, but amount of transcription is low. Activity is primarily focused on completing the DNA replication. Now, each of these phases are highly regulated. Once the genome is replicated means it has completely replicated. Okay? It's not like us. We start something and then, correct, while it is being done, we start another thing. I do very often, don't blame me. Correct? Something exciting. Okay, let's keep it and let's start here. That's it. But here, the checkpoint regulation is very, very efficient. Once, until the complete DNA has duplicated, it won't turn on the gap 2. Okay, so these checkpoints are highly regulated and they are critical checkpoint regulators. So once it has completely replicated, it goes into the G2 phase, and G2 phase again the cell grows, lot of transcription and translation because it's making enough protein for 2 baby cells. Does it make sense? So once you have plenty of cells, it goes into the mitosis, it makes two different cells, and so on and so on. If there is problem in some of the mitotic protein, the segregation, the distribution of the chromosome in 2 daughter cells will not be proper. The human eukaryotic chromosome has some central region. What is it called? Centromere. These proteins are very critical in segregation. There are chromatin modifications, there are critical centromere specific proteins. Okay, these centromere specific proteins maintain the centromeric function. If there is a mutation in centromeric protein, the segregation will be abnormal. Some cells will have each of 46 chromosomes, some cells will have 47, some cells will have less. There is a disease, I don't want to go detail, there is a disease called chromosomal triagenesis. Okay, especially it could be genetic, sometimes it could be people who get late age children. Okay, they eventually can develop Down syndrome. Our body is not made to, correct, have the baby at the age of 50. But we are so busy these days, correct? First try to go to college, then try to make some money, correct? By the time we plan for family planning, it's in 40, 45. Correct? That's not the right thing. Nature has designed, correct, to be reproductive organ. Correct? So in those cases, actually I think it's not a good thing, because when people become little bit older, its normal fertilization ability goes down. So they go for hormone therapy, test tube therapy, and all kinds of stuff. They are nothing other than you are fertilizing an egg and sperm outside. You are actually manipulating the mother's environment with some of the hormones, which can misguide the cellular differentiation. Zygote is formed, but tiny amount of hormonal imbalance can misguide the developmental state. In fact, that's one of the cause, I think, related to abnormal child development, autism, and so on. I know in our community, it's not a big community, there are several people who conceived late stage. Okay, there's a high chance of, if it is in vitro fertilization, there's a high chance of multiple fertilization, correct? People get doublet, triplet, I think there is a report up to 7 child, correct? In those cases, because we use hormones to induce fertilization, but those tiny amount of hormone can misguide the development. I know a couple of babies, they are all fine, but they don't speak. Okay? That's one. I don't want to go to that discussion because these are cell signaling studies. People drink alcohol, people do all kinds of stuff. I also do. I am one of the people, I don't want to be separate. Correct? So there are a lot of things going on, and correct? I got lost. So mitosis. So it's what I'm telling is that, I started with chromosomal triagenesis. Let me finish that, correct? There is certain cases normally we have to have 23 pairs of chromosomes, correct? 22 normal and 23rd pair is the sex chromosome. So male is XY, female is XX, correct? For chromosomal abnormal, correct, mitosis, some guy got X, Y, Y, 2Y, correct? There will be super male. There are a lot of genes, correct, which are male specific. Those will be expressed, and sometimes they will do a lot of crime, they will do all kinds of stuff. We are linked. At the same time, it could happen with the female as well, but also there are certain cases. I think the most often happens in the chromosome 21. Okay, it's called trisomy, and sometimes instead of 2 chromosome 21, child fetus have 3 chromosomes 21. So that's called chromosome trisomy, and this kids will have Down syndrome and many other things. So that's why if there is a certain amount of risk for the family history, correct, once the mom becomes pregnant, after a couple of months, doctor put a big needle in the mom's belly and take some cells out of it, not from the baby, from the amniotic fluid, correct, and count the number of chromosomes under the microscope to see if the fetus has any risk. Does it make sense? So those are some of the stuff interesting. Anyway, so these are abnormal segregation defects. Could be related to mutation, could be related to environment, could be related to age, and many things. Does it make sense? So this is cell cycle. What I want to say in the cell cycle that there are 4 phases. The most important thing I want to emphasize is that cell cycle is highly regulated. There is cell cycle checkpoint. Until one phase of the cell cycle is completed, cell never goes into the next phase of cell cycle. Okay, so these are called cell cycle checkpoint regulation, and P53 is one of the cell cycle checkpoint regulators which controls the G1 and S phase transition. Okay, so DNA replication, mechanism, chemistry, prokaryotes, eukaryotes, all same. And we also, I thought I had another slide. Let's go into this slide. In eukaryote, eukaryotic DNA replication, you have, correct, 46 molecules of DNA. They are linear DNA, and it's much more complex than E. coli. About 6 billion base pairs to copy. So 23 chromosomes, 23 pairs of chromosomes, huge amount of DNA needs to be replicated. Some cells replicate in 24 hours. The tumor cells especially, like the first tumor cells which was discovered or culture in test tube is called HeLa. It's the ovarian, I think it's cervical cancer cell line of an woman, Henrietta La, something like that. Correct? Henrietta La. Okay. So this is our cell line, and I think this was discovered in 1952 or 53 in that range. So the hard genome is still replicated for last 70 years, correct? So during this replication, it's possibly no more hard DNA because it has a lot of mutations. But this cell was taken by a doctor, and he put it in a test tube and put some serum, etc., so that cell is rapidly growing. That's how actually cell culture started. He just demonstrated that you could culture the human cell in test tube. Now it's a routine thing that if you come to my lab, we have 20 different tumor cell lines, some of the healthy cell lines. You can grow them in culture. You can grow them in big clusters, depending on the cell type and so on and so on. Okay? So, HeLa cells can duplicate within 24 hours. It will double up. Where the cell cycle, the S phase is very little, maybe 2-3 hours, correct? So within that short period of time, it has to replicate a huge genome. And the way it does is that it has multiple origin of replication. Origin of replication is, so replication in vivo does not start every place. Okay? It does not start every places. There are certain sequences within the DNA which is recognized by the replication enzyme at the beginning. Does it make sense? So these are called origin of replication. We have studied in Biochem 1 about the plasmid, correct? Plasmid DNA. What is a plasmid? That's not enough. Any circular DNA cannot be plasmid. There is little more than that.
It's a circular extra chromosomal DNA. It's not in the main genome. It's like a site they could use to.
That is true. This is an extra chromosomal circular DNA. But there is something more.
In directions.
Replication. All replication will take place in direction. Something more required to be a plasmid. Did you study something called shuttle vector?
No.
Did you? Did you study the cloning section of the Biochem 1? I'm sure you did. Huh? So any circular DNA cannot act as plasmid. If we introduce this circular DNA, okay, it has to replicate within that particular bacteria or host. Does it make sense? So replication means it needs a particular sequence called origin of replication. Origin of replication, or it is called ORI-C. Origin of replication. This sequence has to be there. If it has to replicate within the bacteria, E. coli, that plasmid must have E. coli specific origin of replication, so that E. coli proteins can recognize and replicate into two plasmids. Without this origin of replication, no circular DNA can replicate in any host. Does it make sense? So plasmid means a circular extra chromosomal DNA, but it must have a origin of replication. If it is E. coli specific, then this plasmid must have the E. coli specific origin of replication. But if it has to grow in yeast, correct? Plasmid does not infect us so easily, because even if we get injected with the plasmid, it will not replicate because it does not have human specific origin of replication. Does it make sense? So if the plasmid has origin of replication or EC of E. coli versus yeast, that means this particular vector, one has E. coli specific and also yeast specific origin of replication. there are two different origin of replication. This particular plasmid can replicate both in the E. coli cells, also in the yeast cells. When we want to manipulate DNA, we want to do a gene therapy. Some protein is mutated or not expressed in certain disease, correct? You want to supply a right copy of the genome using a plasmid DNA or vector, correct? So there, when you want to design your recombinant protein, correct? You want to make sure that this recombinant DNA is able to replicate in human cells. Does it make sense? What you need? You need a human specific origin of replication to be present in that recombinant. Does it make sense? So that's why we say that this protein was cloned in a human specific vector. There are vectors which are specific to E. coli, vectors specific to yeast, vectors specific to human expression construct. That means nothing other than having a human specific origin of replication. If it does not have origin of replication, it's not going to replicate. Does it make sense? So, origin of replication is the sequence of the DNA which is recognized first by the replicating enzyme. There are a lot of stuff which we are not going into the detail. So, for the replication of a huge genome like human, there are multiple origin of replication. So replication starts not only in one site, but at the same time at different sites. Does it make sense? It's like construction of big highway. Correct? If we start one place and one set of people are working, then it will take maybe a couple of years. Instead, you put multiple, correct? Site construction and eventually connect them, it will be much faster. And this is the same concept is used here, that there is multiple origin of replication, one for every 3 to 300 kilobase pair in mammalian cell. So that you start the replication whenever the cell cycle checkpoint signals that now we are ready for replication. All these replicating enzymes binds to this replication origin and starts to replicate that segment of the DNA, and eventually they connect. So within short time, you are able to replicate such a huge genome with high accuracy. It's not just replication, right? It has to be done with high accuracy, and that's the proofreading activity of the polymerase. So in eukaryotes, there are many different DNA polymerase. We are not going to get into the detail. This is a replication, eukaryotic specific replication, origin of replication system. Then you see something called ORC. ORC, that means it's a multi-protein complex which binds to the origin of replication. We are not getting into detail though. Okay? So there are many proteins which control the activation of origin of replication. Okay. So this is the crystal structure I mentioned last week, that this is a E. coli DNA polymerase done by this is not the DNA polymerase structure of the pCNA. pCNA, we are not getting detail in this class. pCNA is a clamp. It's a protein which helps in proving the polymerase. Okay. The polymerase crystal structure was done by Tom Stiles. What is that structure? Yeah, here is the structure which is these are all. This is not the Tom Stiles lab, but another big crystallographer, John Kurian. I heard him his seminar when he was young from Rockefeller University. Big crystallography lab. Okay, they isolate all these proteins, crystallize, and so on and so on. Very nice symmetrical crystal structure. Paul 3. I think Paul 1 was done by Tom Stiles from Yale University. Anyway, so another concept which I like to introduce. So you have a big cell. You have a big cell and you have nucleus. DNA is big. So if this is your DNA, replication is going here. We normally think that DNA polymerase is moving forward until the end, correct? It's replicating at the end. But people like to believe that DNA polymerase do not move. DNA polymerase rather present in certain space, and the DNA is, the thread is kind of pulled. So your machine is fixed here, DNA is feeding from one end, other end it is going out. So DNA polymerase is actually not physically moving, rather staying somewhere, and it's kind of ribbon is being fed and going out with replication. So this is called replication factory. So you are feeding the DNA, going out. Does it make sense? These are some of the basic concepts you want to know. Now I want to talk a little bit of eukaryotic chromosome DNA replication. Now, so far what we learned, how many chromosomes E. coli has? How many chromosomes the E. coli bacteria has? One. How it is? It's a big circle, correct? So E. coli has, anybody knows how many protein coding genes E. coli has? It's about 4,500 or so. How many protein coding genes human has? About 20,000. Not only protein coding genes, there is lots of non-coding RNA, hundreds of thousands, right? So there are these. So in E. coli case, you have a big chromosome, one chromosome circle. You start with one place, you can finish it with multiple origin of replication. You can finish and close, correct? It's no problem. Complete, finish it, seal it. But when it is a human chromosome is not really circle, correct? There are 23 pairs of linear molecules. Does it make sense? So there is your centromere, and the ends of chromosome has a very significant function. Ends of chromosome which are here, correct? End of the linear DNA molecule. These ends of chromosomes are called telomere. Okay. Normally, the ends of chromosome, telomere in eukaryote, there is some repeated sequence, some DNA sequence which are highly repeated. In human, This repeat sequence is, I think, ETA, CTA, GGG. These are called telomeric repeats. Okay? So this is the telomeric repeat present in human and in other organisms. The sequence is slightly different, but they are repeats. And this size of this repeat could vary anywhere between 200 to 3000 repeats at the end of the DNA. If you compare the repeat size, if we take the DNA from a newborn child, correct? Newborn child versus the mother and the grandmother. You are comparing, you are taking the DNA from these three people, and you will see that repeat is very big in the child, medium and small in the grandmother. This is baby, this is mom, this is grandma. Correct? So you see, with age, this number of telomeric repeats are going down. Telomere replication is a big problem. We'll talk about that. So this number of repeats correlate very well with aging. If for some reason, when we are young, these repeats are abnormally lost, okay, it's not replicated properly, efficiently, we lost the repeat, it will be the premature aging. Okay, so always it is important to be healthy, and to be healthy, you need to keep your telomere, stop the telomere shortening. At the same time, if the telomere is lengthening, it's a problem, it's a cancer. Okay, so if the telomere from the large to small, if the telomere for some reason it becomes shortened, cells will die. After a certain threshold frequency of the telomere repeats, cell cannot survive. So telomere sequence, the number of repeats, is critical for cell survival. If the telomere repeats are abnormally increased, cell will not die. It will proliferate. That's the cancer cell. So all the cancer cells has really abnormal high telomere numbers. Okay? So there is a therapy, if you can stop the telomere replication, then it could cure the cancer. So now let's talk about little bit about the telomeric DNA replication, the linear DNA. So we have two strands of the DNA. Let's say 5 prime, 3 prime, 3 prime, 5 prime. We separate, correct? So 3 prime, 5 prime. Now if you have a primer here, correct? Primer here, this is 3 prime end, it will replicate. Does it make sense? Now the first primer is what? RNA, correct? Eventually you degrade this primer. Does it make sense? And this gap at the 5 prime end of the nucleoside strand is not replicated. You need to fill up this gap, you need a primer, correct? But there is no extra DNA where the primer can hybridize. Does it make sense? So there is a problem because primer do not have position to sit. So there is always a problem of DNA replication at the end of the chromosome. Rest is fine, but once this gap is produced, if it is not replicated, correct, next round you lose that part of the end. So every round of replication, if it is not replicated properly, the DNA becomes shortened. Eventually, cells will not survive if DNA is too short. So there is, to avoid this replication problem, we have an enzyme which helps specifically the replication of these ends of the chromosome called telomere, and the enzyme is called telomerase. Telomerase, I think the Elizabeth Blackburn, the name of the lady who discovered the telomerase. I think she was a postdoctoral fellow when she was discovered this telomerase. Okay, so she did not have in her own lab, she was a postdoc, and she discovered that there is an enzyme which are critical in replicating this replication and maintenance of the telomere length. This is called telomerase, and she got Nobel Prize. I don't know the year, I think somewhere in the 90s, for her discovery of a telomerase. And I was reading those days the news that she was going to drop her young baby into the elementary school, and somebody told her that he got Nobel Prize. Okay, so that's kind of exciting. So telomerase is critical for replicating these ends of the telomere, and the way it does that. So you need a primer. That's the problem for end of the DNA replication. What telomerase does, it's a reverse transcriptase like enzyme. Reverse transcriptase it supplies the RNA, makes an RNA which is integrated with the enzyme. This RNA acts as a template to synthesize this RNA-dependent DNA. So what it does, telomerase, as an RNA molecule, it's a reverse transcriptase. That RNA, it acts as a template and synthesizes this repeated end of the primer. Does it make sense? So with the help of this internal RNA molecule, it helps synthesizing these ends of the DNA and maintains the ends of the DNA length. Telomeres could be pretty long. The structures at the end of the eukaryotic chromosome consist of 5 to 8 tandemly repeated G-rich nucleotides. Telomere structure is totally different than the normal double helical DNA. Sometimes you see the quadruplex, sometimes you see the T loop at the end of the DNA. Where is that? I don't have the slides. So the ends of the DNA, if this is your chromosome, telomere sometimes form this type of structure to stabilize. So the repeat sequence, the TTA GGT, which is in human, they could form quadruplex-like structure. It's not double helix. So these unique structure, along with variety of proteins, helps it not to degrade, rather stabilize. And telomerase is the enzyme, it's a reverse transcriptase, which helps to replicate the ends of the telomere. What is this slide? Ends of the so this primer gap is filled by the telomere with its internal RNA. Read a little bit more detail you will get. So what I want you to know that telomerase is the enzyme which is critical for maintenance of the telomere length, correct? And it's a reverse transcriptase. It supplies its own RNA, internally built RNA, correct? Supply the primer and synthesize these gaps. So if telomerase is mutated for some reason, telomerase is not functioning, what will happen? Cells will die, correct? You lose the correct repeat. In fact, as we grow old, lot of oxidative stress or the telomere dysfunction, telomerase activity goes down. So that's why with aging, we have less number of telomere repeats. Does it make sense? So people who are abnormally stressed, correct? Like me, correct? So we get age very fast. So you don't want to be stressed at all, right? Be happy because your telomeres needs to be maintained. In fact, normally if there is a mutation or something, correct? Telomeres is abnormally shortened in the childhood and their telomere activity is low, telomere is shortened, and they prematurely age. Okay, so to avoid the telomere shortening, correct? I will give you some trick. Correct? There is a compound in red wine. Anybody know? Huh? Resveratrol. You already know it. The trick, correct? Resveratrol comes from these red grapes, correct? This resveratrol basically quenches the oxidative stress. If we get angry, stressed, you produce sort of free radicals, and you need to quench these free radicals. Otherwise, they will damage DNA and so on. So basically, you want to take some antioxidant, which is nothing other than, correct, some unsaturated molecules, correct? Resveratrol, polyphenols, like soy milk has a lot of these polyphenols, correct? The colored vegetables, pepper, red pepper, white pepper, they have a lot of these unsaturated bonds. So that's why they are good for health, because if we consume some colored stuff, they take care of our correct oxidative stress and keeps our telomere active. That's why people say that okay, drink a little bit of red wine on a regular basis, especially when you grow old. Don't drink right now. And I did not tell you to drink. Correct? But I just told you the story that yes, if you have to drink wine, drink the red wine because it has justification to supply some antioxidants. Correct? So there are experimental evidence which support that red wine actually helps the telomere function. Okay, but for some reason, if instead of controlling the telomere, if the telomerase is hyperactive, then instead of shortening or maintaining the telomere length, telomere expands. This is expansion mutagenesis. And under those circumstances, correct, the cells would not like to die because telomere is hyperactive. That's cancer. If for some reason telomerase enzyme is activated, it's bad. You can turn a normal cell into cancer cell just by activating the telomerase. There is a scientist in MIT, name is Weinberg. he actually discovered the trick of how you can convert a normal healthy cells into cancer cells. That's not what we want. Right? We want the other way. Other way is much more difficult. Right? How you convert the tumor cell into normal cell. There is certain missing keys how to tumor, and maybe some of you. So telomerase is very, very critical, and that's all I wanted to talk about eukaryotic replication. That we have a lot of replication origin, we have a cell cycle, cell checkpoint regulation, then more little bit about the telomerase and telomere. So what is a potential therapy for anti-cancer therapy based on what we discussed on telomerase? So if you can inhibit the telomerase activity, correct, which is hyperactive in tumor cell, you should be able to control the tumor growth. Correct? So there are some molecules, antisense, oligonucleotides, which has been developed to knock down the telomerase level, and those are good for therapy, cancer therapy. Okay, so with that, we want to briefly touch base about the viral genome. Some of the virus, their genome is nothing other than a RNA molecule, correct? RNA molecule, viral genome, which is genome is capped with some of the capped proteins, which are useful for recognizing the host cell surface, correct? Once the virus recognizes in host cell, it injects the RNA into the host cell, and this RNA acts as genetic material. It's duplicated. It makes a complementary DNA. With an enzyme, what is the enzyme? So this RNA acts as template and makes the DNA complementary to this. This DNA has few critical protein or enzymes which are required for viral replication. Does it make sense? So this conversion of this RNA into this hybrid RNA DNA RNA hybrid is critical because you are making a stable DNA, correct? So what is the name of the enzyme? It's a RNA dependent DNA polymerase. Normal DNA polymerase requires DNA as a template, correct? This is a RNA dependent DNA polymerase, and this is called reverse transcriptase. Actually, this telomerase is a reverse transcriptase because it uses RNA as a template. So this is RNA dependent DNA polymerase, which is also called reverse transcriptase. Why reverse transcriptase? Transcription means DNA to RNA, correct? So RNA to DNA, that's why it is called reverse transcriptase. Does it make sense? So this is reverse transcriptase, and this reverse transcriptase helps to convert the genome into DNA. Then there is something called RNase H. RNase H, which degrades this RNA to eventually produce the DNA. And this DNA is transcribed and so on to make the new virus, package into new virus. So, one of the most promising therapy for, what's the time? Are we on keeping the time? It's 10:40 now? Okay. So we still have 2 minutes. So RNase H degrades these RNA, so you have DNA, and that is now acting as template for transcription, making the viral proteins, packaging, and so on. So, what is the therapy for this HIV? Correct? There are many viral therapies follows the same path. So, one of the most promising therapy is if you can inhibit this reverse transcriptase. Correct? This particular compound called AGT, azidothymirin, it's like a nucleotide analog. Does it make sense? It's a thymidine analog, and this thymidine analog, if it is added, it inhibits the reverse transcriptase activity because it gets incorporated into the DNA. Does it make sense? And there is no, for the DNA replication to happen, you need what? What do you need? What is the requirement for DNA replication? Your primer 3' hydroxyl group has to be free. And in this case, instead of a hydroxyl group, we have this azido group. Once it is incorporated, replication is stopped. Does it make sense? So that's why this molecule is a drug. It's a reverse transcriptase inhibitor. But most of the time, the virus is very smart. Correct? Once they treat with the drug, they do something and they make new virus. Like new type of virus, mutation, rapid mutation. That was one of the major problems with HIV. So, what is the most useful therapy there? How it is controlled? So, the widely used therapy is not just one drug. Just give a cocktail of drugs targeting different pathways. Right? So, it suppresses the viral replication. In fact, the therapy which is used for HIV treatment may also be used for other viral therapies because they are similar. Does it make sense? David Baltimore from MIT actually discovered this reverse transcriptase. The reverse transcriptase has the RNA directed DNA polymerase activity, RNase H activity, which degrades the RNA from the RNA DNA hybrid, then it can also replicate on DNA. HIV therapy, azidothymirin. Correct? Does it make sense, some of these concepts? I think that's all I wanted to say on the replication. Okay? So basic mechanism of replication in E. coli, basic chemistry of replication, proofreading, then E. coli replication fork, what are the enzymes you need, then you go to the eukaryote cell cycle, correct? Origin of replication, and telomere replication, and then viral DNA replication. Does it make sense? Read this. Next class will briefly touch recombination and repair. Correct? That's a big one whole semester one could spend on talking repair and replication. Recombination. But we will finish in one class. That's introduction.